Thank you very much, Professor, for your kind introduction. I'm Jie Hu from National Taiwan University and National Yangming Chiaotong University. And today I'm sharing our research topic, single-cell multi-omics reveals the regulatory networks in neuroblastoma cell differentiation. Okay, and we have no conflicts of interest to disclose. First, neuroblastoma, or NB, it's a very common cancer type in children, and it originates from neurocrest progenitor cells. It got some subtypes for NB. For example, MECAN amplify and non-amplify. MECAN is an oncogene that correlates with rapid proliferation, and the cell lineage of NB, neuronal, mesenchymal, or intermediate. And there's a drug, O-trans-retinoic acid. It derives from vitamin A, and previous study shows that RA actually induce neuronal differentiation on NB cell in vivo in zebrafish and in vitro in cancer stem cell plates. Okay, so here's our question. What is the molecular mechanism governing ATRA-induced NB cell-fate reprogramming? Or how does neuronal differentiation occurs on cancer cell? And here's our study workflow. First, we validate the efficacy for ATRA-induced NB cell differentiation. Then we conduct single-cell ATAC-seq and single-cell RNA-seq. We integrate the data and then perform DEG, differential express gene analysis, and pathway enrichment analysis, LS transcription factor prediction, predicting the key regulatory elements for this effect. And here's our result. First, we validate the efficacy of ATRA. As you can see, the neuronal differentiation generates neurites, and the neurite count correlates with the concentration of ATRA gradually from left to right. And in both kinds of different status of MECAN amplification, on the left side, it's a MECAN amplification cell lines, and on the left side, it's the MECAN non-amplified cell lines. And then we conduct single-cell multi-omic, include ATAC-seq and RNA-seq. ATAC-seq investigate the chromosome accessibility, and RNA-seq investigate the gene expression value. And we incorporate the data by charade package, and with some different cell lineage markers and cell type markers in previous study, we annotate the cell type back onto the data. And as you can see, on the left upper side, on the right upper side, it is the UMAP. And on the upper left, it is the MECAN amplified cell lines. On the lower right, it's the MECAN non-amplified cell lines. Okay. And then we perform differential express gene analysis. And as a result, we find many genes differentially expressed after ADRA drug treatment, including cytoskeleton remodeling and retinoic acid receptor regulating. But interestingly, their expression oppositely patterns on the amplification status of MECAN. For example, if one gene is highly expressed in the treatment group of the MECAN amplified, then it is lower in the MECAN non-amplified treatment group, and reversely too. Then we want to ask, what does these genes involve in biological pathways? So we conduct biological pathway enrichment. And as the same, on the left side, it is the MECAN amplified group. On the right side, it is the MECAN non-amplified group. And many biological pathways involved in these important groups are inversely, oppositely expressed in MECAN amplified and non-amplified. So here's our first summary. First, ATRA induced effectively neuronal differentiation in NB, and many genes function in cytoskeleton remodeling and retinoic acid signaling. And more importantly, many pathways involved in embryonic development, cell lineage specification, tissue differentiation, and retinoic drug catabolism. But these pathways and genes oppositely express between MECAN amplification status. Okay, so the part 2 of our result, we want to ask, what is a key regulatory element for this gene regulatory network? And here we convey the service of Cynic Plus, a package. And Cynic Plus cements the transcription factor expression and the chromosome region accessibility and the downstream gene expression into a whole component and calculate its fold change and its significance. And here's the description below in the citation. And here's our result. So how do we read it? There are many different TFs involved in different gene regulatory network are enriched after ATRA treatment in all different cell lines. But we can induct them into 3 different groups: the green one, the orange one, and the red one. The green one are the RA receptors, the retinoic acid receptors groups. They are highly expressed after ATRA treatment within all groups. And the orange one are Hox genes and morpho genes controlling development. They are both highly enriched after ATRA treatment. But the red one, they are cancer stem cell markers and oncogenes. They are decreased and inhibit after ATRA treatment. And after we calculate the significance and the fold change of different irregulons, we differentiate 2 key elements, KLF2 and KLF6. KLF is Kruppel-like family, and they are Hox genes. These 2 are the key regulatory elements in MECA non-amplify and MECA amplify. So here's our second summary. The gene regulatory network of our receptor are activated. The morphogenes and Hox gene regulatory network are activated too, but cancer stem cell markers and oncogenes are suppressed. And we find 2 key regulatory elements, KLF2 and KLF6, expressed differently between MECA amplification status. And the last part of our results, we want to validate that this shows in patients. So we conduct survival analysis on these 2 key differentiation regulators, KLF2 and KLF6. And as a result, here we use the R2 neuroblastoma website, and it has patients' data with different gene expression in the patient. And as you can see, when KLF2 and KLF6 and their downstream genes are highly expressed, better patient survival rate and survival time is performed, mostly, except for GAL, this downstream gene of KLF6. And the patient is more than 400 patients, so it is significant. So at last, we have our proposed hypothesis for this ATRA-induced MB differentiation effect. The key regulatory elements are KLF2 for MECA non-amplified and KLF6 for MECA amplified. And they have some downstream genes to conduct the differentiation and neuronal differentiations. But more interestingly, KLF2 have oppositely regulate 2 different complexes of RA receptors, retinoic acid receptors, CRAB2 RARB and FAB5 PPARG. And they similarly opposite regulates MECA. One activates MECA and one decrease MECA expression. So this forms a negative feedback loop. And this might explain why we see different patterns, especially opposite patterns in the DEGs, in the biological pathways, and the TF enrichments. So here's our proposed hypothesis for wet lab validation. And we have done some 3D organoid experiments, and we will do some spatial staining and imaging in the future to look at the organoids. So here's our conclusion. First, we conduct single-cell multi-omic analysis and reveals KLF2 and KLF6 Kruppel-like family induce ATRA-induced neuroblastoma differentiation. And KLF2 and KLF6 gene regulatory network express oppositely based on MECA amplification status. Also, many TFs and genes are identified in this process for future validation and possible candidates in drug repositioning in neuroblastoma differentiation therapy. But I think more importantly, these TFs and genes mostly function in morphogenes, Hox gene, and cancer stem cell repressors. And this might be a generalized suggestion that maybe in other cancers, especially those cancers affected by MECA and those cancers affected by stem cell-like progression and cancer stem cell, we can use similar process or similar concept to investigate and possibly to treat these different cancers for better patients' well-being. And alas, my acknowledgement. I thank my professor very much, my advisors, Dr. Ranshi Fen and Dr. Shenzhen Huang, and Dr. Qiaolong Xu for the Single Cell Center in Taiwan, Dr. Chao Wei for the cell culturing, and Mr. Jiang Sun for teaching me the coding for the analysis, and every lab members in our lab, and NTU and NYCU, and our fundings. Thank you.
Very nice work. Yeah, thank you very much.
Thank you.
Very interesting. So are there any small molecule inhibitors that can potentially target those identified master regulators to see if you can achieve some efficacious, treatment, improvements?
Yeah, this is a future direction we're working on, like maybe some knockout or some inhibitor that can perform some well-lab evidence. Yeah, thank you very much.
So any questions from the audience? So your wet lab collaborator, you already developed developed the organoids, test the organoids.
Yeah, we're working on it now, like right after this summer. And some preliminary data shows very exciting results. Maybe we can share it next time in the future works.
Yeah. Very nice.